All right, guys, here we go. Video number 2. We're going to get right to it. So complex inheritance patterns. This one is complex. So it's not something that we're probably going to go all the way through today. Maybe it's going to go maybe max 30 minutes. Sometimes it go longer than I want to, like on the last video, really. But we needed to, I think. So we'll see how long it goes. But these are complex inheritance patterns. So they're different than just dominant and recessive. Before it's real simple. Hey, if you got the dominant gene, you express the dominant gene. I mean, if like pea color, right? If you had big A and little a, and big A stood for green, little a stood for yellow, well, you were going to be green, right? That's as easy as that. You still have the yellow gene, but you don't express it, so you don't see it. Well, it's not always that simple. It's not always that simple. There's more to the story. What if you had a purple gene, or excuse me, say you had a white gene. This is a simple example. Say you had a white gene and a red gene. And what if they're both dominant kind of together and you got a red and white spotted plant? Or what if you had red and white and they were both incompletely dominant and you got a pink plant? So that's the case. And we'll go through some of those today. And we call it codominance, incomplete dominance. And so it's not just dominance, law of dominance. So there's more to the story. And there's more than that, right? Okay. And so let's do it. Here we go. All right. So complex inheritance patterns. Boom. Okay. Review. Whoa. Review of Mendels Laws. We had the law of dominance, which that's what we went over yesterday. That was monohybrid crosses that we did yesterday, and and what we did today was the dihybrid cross, which is a case of dominance. The law of dominance. Some alleles are dominant to others. Okay. And then we had the law of segregation. The law of segregation just simply states that each organism inherit 2 copies of each allele, one from mom, one from dad, right? That's what we're saying, right? You get 2 copies. You got 2 copies of every gene that you have. And so you got one for mom, one for dad. That's the law of segregation, okay? And then the law of independent assortment was that genes for different traits assort independently during the formation, that says, of gametes, sperm and egg, okay? There you go, of gametes, sperm and egg. So what does that mean? That means if you get, say, like a dihybrid cross, let's say we got a gene for pea color and pea shape. Well, let's say you get the dominant allele for pea color. You get green. Well, that has no bearing at all on the shape of the pea. You're not any more likely to get the dominant pea shape, like round, okay? You're just, it doesn't matter. There is independent assortment. They're independent of one another. So whatever gene you get for pea shape doesn't affect what gene you get for pea color. It's equally likely, okay? All right, and this was Mendel's famous experiment right here, guys. You know that experiment. He took 2 pure breeding plants. I mean, like this one only gives you yellow peas. This one only gives you green peas. And then he gets the F1 generation where one of the colors, don't let the colors confuse you so much. It's basically saying, hey, this one, in this case, we're saying yellow is dominant to green, okay? What actually is the case doesn't necessarily really matter. It matters as long as we understand the concept, right? To this case, like the green went away. But all of a sudden, he took what the 2 of the offspring and bred them together, and then all of a sudden, the green came back. So that led to the idea is it must be a recessive trait. And guys, this was in the 1800s, by the way. Mendel did this in like 1850 or something. Okay, he's figuring this stuff out maybe later, somewhere around there. Okay, yeah, I mean, this is in very long time ago that somebody could figure this stuff out. It's pretty incredible. Okay. What did I just do? Okay. Let's make that. All right. So, okay. So exceptions to Mendel's laws. Some alleles are neither dominant nor recessive, and many traits are controlled by multiple alleles or multiple genes. Think about your own traits. Which do you already know don't fit within Mendelian genetics? Think about skin color. Think about how many variations there are of skin color. Think about eyes. There's not 2. There's not just like brown and blue. There's not that, right? Look at these kitties, right? There's so many different variations on their fur color. So that's one thing. Observation leads us to think about things, right? It's like this can't be everything, right? It can't just be two traits that determine everything. There's more to the story, right? And some things get really complicated with genetics. There's some traits that are determined by more than one gene, right? So it can be polygenic inheritance, where you have a bunch of genes that determine a trait. Okay. All right. So the first one we're going to talk about is incomplete dominance. Okay. All right, so incomplete dominance. And we have this case of these beautiful roses right here. It says incomplete dominance is the heterozygous phenotype is somewhere between the two homozygous phenotypes. Okay. It says neither allele is completely dominant or recessive. So that's why it says incomplete dominance. So what that means is what, like it was the example that we gave at the beginning of this. If I take a red rose and cross it with a white rose, I get pink flowers. That's pretty cool. Okay, red and white makes pink. They kind of mix together. That's called incomplete dominance. And what you're going to see is incomplete dominance is really easy to get confused with codominance. Okay, so there's dominance, which is what we already discussed. Now there's incomplete dominance. And in a second, we'll see codominance. All right, so let's do an example of incomplete dominance. All right, incomplete dominance. So it says in humans, curly hair, which is going to be, and so guys, the letters we use, how you write this stuff doesn't really matter. Again, it's notation. It's like when we're doing math or something like, and it says you use A to represent a variable. Well, you could use B or C. It doesn't matter really. It's just a letter to represent something. So in humans, curly hair, which is going to be represented by big H, is incompletely dominant to straight hair, which is going to be that phenotype, the physical characteristic of straight hair, is going to be represented by the genotype H asterisk H asterisk, okay, or called asterisk, okay. The heterozygous individual has wavy hair, okay, so big big H asterisk, because they're not, we can't use a little h because that was telling us we already used that notation for the law of dominance, dominant and recessive. So we can't use that with this. We just use something else. Okay. So it says cross two people with wavy hair. Okay. You do that. Let's do it. All right. So two people with wavy hair. So here we go. We got big H, asterisk, big H, asterisk. Okay. So big H, big H, big H, big H, asterisk, big H, big H, asterisk, H, big H, asterisk, big H. Almost there. Asterisk. Okay, good. All right, so now it gives us a question. This is how you answer these questions a lot of times. It'll ask you this question. It says, okay, you did this, great. Now it says, what's the genotypic ratio? Let's go through and look at this. There's four possible things here, like four boxes, 1, 2, 3, 4. Okay, so let's see. This person is going to have curly hair, right? Okay, curly hair, because it says big H is curly hair, and it's incompletely dominant to straight hair. So we have one that has curly hair, one that has straight hair. So curly, straight, right? Because it's big H asterisk, big H asterisk. And then we have two that have wavy hair, wavy. Pretty cool. So when it asks us what's the genotypic ratio, well, one has curly hair. Let's put a little colon. Two of them, have a wavy hair. Gosh darn it. So one has curly, two has wavy, and one has straight hair. So the genotypic ratio is 1, 2 to 1. Okay? So for every 2, so you got basically a 50% chance that you will have a child. If this is the case, this is parents, 50% chance you'll have a child with straight hair. And you have a 25% chance that they'll have curly hair and a 25% chance that they'll have wavy hair. You got straight hair. Gosh. You got all the 3 possibilities of hair. Pretty cool if that was the parents. And the phenotypic ratio would be the same because there's 3 different possible phenotypes. All right, we just said it. So the phenotypic ratio will be the same thing. That's not always the case. The phenotypic and genotypic ratio are not always the same. So 1, 2 to 1. 1, 2 to 1. Okay? Awesome. All right. Okay? All right, so next one is, what about roses, these pretty roses here, okay? So how about I just talk through this one? How do I clear the board? Oh, there it is, clear screen. Okay. All right, so Fitz loves growing flowers for his friend Olivia. That's kind. Her favorite flowers are roses. And roses are found in red, big R, blue, big R asterisk, and purple. Okay, which is big R asterisk. All right. So we had red, blue, purple. All right. What would happen if Fitz crossed a blue rose and a purple rose? Okay. What would happen if you crossed a blue rose and a purple rose? All right. All right. So we got blue is here. It's big R here, big R here, make our little things there, with a purple rose. We have purple is big R asterisk. Okay, so let's see what is he going to get here. What is he going to get? We can write it. Oops. Here we go. Big R asterisk, 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 big R. Okay, hopefully you see where I'm getting these things from, right? I put this one here, this one here. It doesn't really matter which comes first. This one here, 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 this one here. Okay. All right. So what do we have, guys? We've got a purple rose here, right? We've got a purple rose here, and then we have a blue rose here and a blue rose here. No red roses. Okay, so if you cross blue and purple, you're not getting any red from that. Okay, so it says Fitz loves growing flowers for her friend Olivia. Her favorite flowers are roses, and they're red, blue, purple. What would happen? Okay, and then that would tell. So the genotypic ratio would be basically, okay, we could put it as 2 to 2. We want to simplify it, right? 2 to 2 would simplify to 1 to 1. Okay, so it's 1 to 1. You have 50% chance of getting purple or blue or purple. And the genotypic ratio would be the same. It'll be 1 to 1 or 2 to 2. So you have the 2 to 2 should be simplified to 1 to 1. Okay? Good. Oops. All right. Codominance. All right, this is what I was talking about. This is typically easy to get confused with incomplete dominance, but what about this beautiful orchid right here? Right? How do you get those? Well, you get both phenotypes expressed, or both, should we say, both genotypes expressed as phenotypes. You don't get them. You don't with codominant. You don't get a blending. Right? Red and white don't make pink. You get a red and white flower. Right, where they both show up. Okay, so that's codominant. They're both expressed. They're codominant. They're both shown. Both phenotypes are shown. Okay, a cool example of this is your blood type. We'll talk a little bit about blood type here in just a second. But blood type is an example of multiple alleles as well, where there's more than 2 alleles. There's 3. So right now I'm just seeing 2 alleles for each type. Right. Okay. So codominance. Let's do an example of codominance. Again, I'm probably going too fast for you to write everything on this slide, but trying to keep this as short as possible. The videos don't get too long. So you could always pause it here, write because this is definitely something. Obviously, it'll be on a quiz. Hey, you still could have quizzes, right? Take good notes. You get to use your notes like always. Um, so make sure you're doing that. Okay. Ooh, an Appaloosa horse. Beautiful horse. So Appaloosa horse is an example of codominance. Pretty cool, guys. Pretty cool to see some of these real world things, like these flowers and like Labrador Retriever coat color, which we'll see in a second, which is actually kind of complicated. Um, but uh, but some things are simple, or relatively simple, I guess. All right, so let's do codominance here. And again, you kind of see different notation here with two different letters. All right. So in horses, brown horses, big B, are codominant to white horses, which are big W, big W. The heterozygote, which now I use two different letters here for this codominance notation. The heterozygote, big B, big W phenotype is an Appaloosa horse, a white horse with brown spots. So cross a white horse with an Appaloosa horse. Let's see what we get. All right, white horse. We have W with, what am I doing, guys? W with a Appaloosa horse, which is going to be BW. Okay. So what do we have here, guys? We have BW and Appaloosa. We have a white. We have Appaloosa. And we have a white. All right. So what's our genotypic ratio? The genotypic ratio is 1-to-1, BW, 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 BW. So it's 1-to-1, so 2-to-2, really 1-2, 1-2, same thing. Simplifies to 1-to-1, like we said. So 1-to-1. And then the phenotypic ratio would actually be the same. So we have a 50% chance if we cross these two horses to get an Appaloosa horse, and a 50% chance to get a white horse from the offspring from the mating of these two horses. Okay, 1-to-1. Okay, cool. All right. Oh, blood type, cool. All right, so these are red blood cells right here. Red blood cells. All right, so blood type is an example of codominance and an example of multiple alleles. And multiple alleles is just saying you have more than two alleles for a trait. You have A, B, and O. O, you said that's not an O, that's an I. And it is an I, but we use a lowercase I to represent O. And the reason it's a lowercase I is the lowercase is showing, right, the dominant and recessive, right? So A and B. So blood types is complicated because it involves a lot of stuff here. So what it is A and B are codominant. Okay. So codominant means both are expressed, right? Both genotypes are expressed on the surface of these red blood cells. I is recessive to A and B. Okay. I is recessive to A and B. So that means if A and B are there, there's no I. And basically what we're saying with I is if A and B are absent, then you represent O with the letter I. So blood type, here it is down here. Your genotype would be II, right? And that's O. That's blood type O. Okay. You could represent blood type A can be represented as AA or AI. It's still A. Okay. And blood type B can be represented as BB or BI, which would be B. Okay. So what we're saying here, A and B are codominant. They're both fully expressed. A and B are both dominant to O. Okay. All right. Let's do an example. All right, guys, you do not have to learn this chart to be cool. This is something like you'd learn in like nursing school. Really, it's just a cool chart to understand. I think you might should understand it, but I'm not going to make you learn it. It's not going to be on a test, say. But it is kind of cool to know, hey, if I have blood type O, okay, if I have blood type O, I can give to anybody, which is pretty cool, right? I can give to anybody. You're the universal donor, but you can only, I'm going to put this somewhere. Anyway, let's put it over here. You can only receive type O, okay? So you got to be careful when you're blood type O that you only get O blood, or it could be fatal. Your blood could clot, right? So one good thing about AB, if your blood type is AB, well, you can get any blood because you're good to go, okay? And it's slightly more complicated than that, but that's the gist of it, okay? And for us, it's pretty cool to understand. So this is a pretty cool chart to kind of get into and try to understand, okay? But again, I'm not going to make you learn this chart on a test. You're not going to have to write it on a test, but you will have to do a Punnett square on a test. Let's do one of those on the next slide. Okay. Example. And sometimes for blood type, you have to draw more than one Punnett square. Okay. All right. You have to draw more than one Punnett square. I know this is a lot, guys. We're going to practice this. We've got sheet after sheet to practice this. We have activities to practice this. There's some things we can do to make sure we get this stuff down. We're kind of hitting it all now. When I get back, we're going to go back to the very beginning of just law of dominance and work through that, work on multiple alleles, work on dihybrid cross and all these other ones. Okay. All right. So if a mother has type AB blood, so the mother has type AB, and a father has type B blood, what are the possible blood types of their offspring? Okay, this is why you have to do two Punnett squares. All right, so the mother is simple. She's type AB. All right. Well, then it says the father has type B blood. Okay. Well, the father has two possibilities. Okay. We don't know. We got to say that. Well, the father could be. Remember, it could be big B, right? Okay. He could be homozygous. He could have gotten a big B from his mom and a big B from his dad, or he could be B O. Remember, or you know what we're going to do, guys? We're just going to write the letter O. Heck with that I. Let me just write O. Okay, I don't think it matters. Okay, I think that makes a lot more sense. So I don't know why the convention is to do an I. I'm sure it's got good reasons, but we're just going to put an O. It's blood type O. Okay, all right. So you got AB for mom, AB for mom. So the reason you have to do two Punnett. There we go. The reason you have to do two Punnett squares here is because dad has two possible genotypes. Okay, so when we do this now, we fill it in like we always have: A, B, 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 A, B, B, B. Okay, A, B, 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 A, O, B, O. Okay, do it just like you always would have. So again, the reason we did two Punnett squares is because dad had two possible genotypes, because it just said he was type B blood. He could be BB or he could be BO. All right. Whoa, last one here, guys. This is the last slide we're going to do today. Last slide we're going to do today, and then we're going to shut it down. We'll do the rest. Look at the next slide. Then we'll hit it. Okay, so stay with me. Here we go. All right. Oh no, the hospital has made a big mistake. This is why these are kind of cool problems. Okay, the hospital's made a big mistake and lost track of which baby belongs to each set of parents. Can you help them fix this before they get sued? Oh my goodness. All right, so the parents, Mr. and Mrs. Allele, they're AB and AB. Okay, so they're easy. They don't have any, just simple what they can be, right? AB, AB. Okay, let's see what babies they could have. They could have a baby that's AO. What am I saying? AA, excuse me. AA. They could have a baby that's AB, A, B, or a BB. All right, so they could. So we know well. We know right now, right? If we look at this, okay, it says baby one has type O, baby two is type AB, baby three is type A. So baby one and baby three cannot be Mr. and Mrs. Allele's child, right? The only even possibility doesn't necessarily mean this is definitely theirs till we do these over here, right? But it's a decent possibility that baby two is theirs. Baby one is definitely not. Baby three is definitely not. So I guess if we know that these are the only three parents, these are the only three possibilities, then the only baby that could possibly be theirs is baby two. So let's just put baby two by these guys, and this is the allele family. All right. And then it says, all right, Mr. and Mrs. DNA, their blood types are O. So that's we already figured this one out. No, we hadn't yet. Never mind. O. And this one is AB. Actually, we have figured this one out. Never mind. All right. So just by doing this, guys, we know right now that this baby, AO, BO, AO, BO. Okay. All right, guys. We know now. Because you can look at this, guys. All right. This baby one cannot belong to this family. This blood type has to be OO, right? If you're O, you have to be OO. Okay. So what it's going to be, guys, is baby three has got to belong to this family, right? But let's just go ahead and finish it up. Baby three is going to be Mr. and Mrs. DNA. And then baby one, if we do Mr. and Mrs. Gene, it's B. What we know already, guys, is that now look, this is saying this person could be BB, but they could also be BO. And we know that this person down here, if they have blood type O, has to be OO. That's the only way you can be O is OO. So BO, OO, there it is. OO, there's baby. BO and OO. Okay. And the reason you have another Punnett square is you could do it as BB. But if you do it as BB, you're not going to get any blood type O's. Okay, you're just going to get BO, 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 BO. So it's all blood type B. That wouldn't work. Right? So baby one belongs to Mr. and Mrs. Gene. All right. Okay, guys. So let's see. We're going to, a good time to stop would be right there, I think. And we'll get back to multiple alleles tomorrow. So you focus on these for right now, and I'll, if I don't have a worksheet for you for this yet, I'm gonna get another one. I think I'll probably text and get another one printed off for you guys, so you can have something to practice along with these notes. And you can always get your Chromebook out and watch these notes back, right, as you're doing the worksheet if you're not watching it as a class. Okay, of course, stay on task and do what you need to do, guys, and rock and roll and learn this stuff. This is some cool stuff. A lot of things you can actually apply in real life. Okay, figuring stuff out. All right, I'll have a great rest of the day.